In this PowerPoint, I will be discussing the roles of qualitative and quantitative data, how they are obtained, what are the types and questions that can be discussed through the different types of data, the benefits and consequences of each type, and also including the issues that can occur. Qualitative data. This is where the information is presented as opinions or viewpoints, mainly to discuss and understand the reasoning behind why the subject of the research has done a specific thing or made a specific decision. This data is where the researchers tend to look at patterns in recurring words to gain a clearer understanding between reasoning and developing their understanding. The qualitative data also is used in small samples because the investigation is very in-depth. As a result of this, the data will be small-scale research because otherwise it would be time-consuming and would lack quality. The information is subjective, yet is extremely rich in quality. This would mean because the qualitative data is very in-depth to opinions and viewpoints that it allows the investigation and research to be discussed in more detail and more in depth. For example, a question which will be used in qualitative data is how does injury mentally and psychologically impact a centre forward? This would be a very good question for qualitative data because it discusses the reasoning behind why the centre forward in football is getting affected and also how it's affecting them, whereas quantitative data would normally just put facts and figures towards it. Examples of qualitative data and how to obtain this type of data is found in interviews, group and focus groups, and surveys. Interviews are one-to-one talk which discusses what the researcher wants to find out and is very effective. Now we're going to talk about quantitative data. This is where the information is presented as statistics, facts, or numerical results. In sport, this is the most popularized type of data as it puts a figure onto the performance of the athletes and it doesn't really focus on why they are doing things, which qualitative data would do. The researcher is aiming to produce unbiased results that can be generalized into a wider population and act as an average. This would be more unbiased than qualitative data as you can't really lie about solid stats and numbers from scientific lab tests, where qualitative data you could alter the results by altering the questions. As it says, the information obtained is objective but lacks detail and depth because all it is focusing on the statistics and not the reasoning why the statistics are like that. For the test, the environment is specifically set up for results to be gathered. So lab tests are in a controlled environment where the subject of the test is put under control variables to prevent any external variables from affecting the test to get the results which are needed and are wanted. An example of quantitative data, a question would be, how does injury physically impact a centre forward's performance? This, if it was qualitative data, it would be around his emotions and reasonings behind how it affected a performance, but this would be how it has affected, physically impacted the centre forward's performance. So this would be looking at his goal rate, his movement off the ball, his movement on the ball, and his speed and other scenarios like that. Quantitative data can be obtained through lab tests and questionnaires. Now we'll be talking about issues when researching. The first issue would be validity. This is a major issue as it could completely affect the question in which we are looking at, and the results can be completely altered by a sort of issue in this validity. As mentioned by Adams in 2010, this is whether you are measuring what you're supposed to be measuring. Internal validity refers to the research findings and how they match reality, while internal validity refers to the extent to which the research findings can be replicated to other environments. That's by Palicio in 2008, and this shows how the internal validity is, if they can control it, and how can that result affect the result, and the external of how it affects the environment, such as the weather and other people in general public. If the researcher has not controlled a variable, it could potentially cause the outcome, which affects the whole investigation. So if a researcher is testing sprint speed and cannot control distractions which could distract the person getting tested, that could completely affect the results of the participant, which would cause the whole investigation to be affected and impacted negatively, and would lead to incorrect results and measurements. A sporting example would, like I said, would be an investigation of sprint times for a 100-meter sprint who researched into. Would the results be about the sprint times and what the research is actually based around, close to the subject? If the results were not close or similar to what was supposed to be measuring, then the validity is low. This would impact the investigation as it would provide us with an incorrect final value. So you want to be as close to the research target as possible and sticking to as close to the subject. Another issue whilst researching would be reliability. This would be if the test was taken again, would the same results occur? In order to carry out the investigation in a reliable fashion, the research must be done the same way throughout the entire investigation. This can impact an investigation majorly, as if an anomaly occurs that could make a spike in the results, resulting in an incorrect measurement and result at the end of the research. There are two types of reliability. Internal, this is the consistency of the results within the test. So if the test was done again in the same conditions by the same person, all the same, would it be the same? Obviously. And external, this is the consistency of the results if the research was carried out in a different environment. So in, say, if a test was carried out looking at the sprint times at colleges for 16-year-olds, for internal, this would be keeping the same test results and same test methods for the 16-year-olds in one college, and external would be the consistency of the results if the research was carried out in a different college. This would affect the validity and the reliability as a result of this. A sporting example would be if a test was carried out into the timings of a marathon run, if that run would be taken again under the same conditions and the same research, then would we expect the result to be near enough the same? If the sources are unreliable, it will impact the investigation by causing the answers to be off subject, maybe an anomaly, or even just incorrect answers in general. An anomaly would be a major fault in a test, as it can give us incorrect readings and affect further tests, and it could affect the performer who is doing the test, as it could affect their performance in the future. Accuracy and precision are major parts of keeping your research on target and making sure that the research is getting the answers that you need to get and as close to the target and the final value as possible. Precision is gained through a thorough and well-kept systematic approach to find a result which we are looking for during our research. The better the systematic process, the more precise our results will be. Accuracy is how close your findings are to the true value. The higher accuracy you get, the closer you are to the target you are aiming at. So the target in this case would be the final answer that you're looking for and the final bit of data you want for your results and your research. A way to portray this accuracy and precision is with the archery method, which is shown in the top right hand corner of the slide. Along the y-axis is the precision, and along the x-axis is the accuracy. As precision is collectively how we are getting our results to be accurate. So this would be if your results are accurate but are done through a less precise method, then it will be shown on the bottom right of the diagram, so the low precision, high accuracy. So it's around the target, which is the middle white dot, but it's around it, but as it's not precise, it's not bang on every time, and it will lead to the results being spread out over that target we want. If you have low accuracy but high precision, then all the results are going to be bunched together, as you can see in the top left diagram of the archery. They're away from the target, so as the accuracy is not there, but the precision is there, so you're doing the same systematic method but away from the target. This would lead to a false reading of the research and could affect the research in the near future. Low precision and low accuracy would lead the results to be spread out away from the initial target, so the results would be altered and affected negatively, and the results would be unreliable because they have low precision and low accuracy. High precision and high accuracy would be shown in the top right archery board, and this is where your precision and you're doing the same method every single time spot on, and your accuracy is perfect, so it's getting bunched up collectively onto the middle target where we are aiming to hit, and it is the perfect method. If you can obtain high precision and high accuracy, then your method would be the most reliable and the most close to the target as possible, which would be perfect for anyone doing any type of research. We'll be talking about the types of data now, with the two types of data in focus, which are primary data and secondary data. Primary data is the type of data which the researcher finds out for themselves, and they find it out firsthand. They make sure the data is unbiased and is spot on to and subjective to what they are looking at. This is beneficial because you can make sure the data is true and unbiased, yet if the researcher is investigating into something they are passionate about and one-sided about themselves, they may take a one-sided view, which makes the investigation biased as a result of that. Primary data is by far the most effective type of data, as you can find what you need to know for yourself, and you can make sure that the results are true for yourself without people making them up. And even though it is more time consuming, as you have to do the test yourself and do the research yourself, it is more reliable for your research. Secondary data is the type of data which is used in research when the researcher has found data either online in journals or reports from anywhere else. This can be found in books such as sports science books, and this is less time consuming but less reliable at the same time, as you don't know where the data has been coming from, you don't know if the data is true and reliable, and also you don't know if the data is what you are looking for exactly, whereas primary data is you can narrow down to what you're looking for. However, secondary data, it may be a more spread out research which has bits of data that you might take out of it which is out of context, which could affect your research negatively. This data can be beneficial because it can be from a mutual point in an argument, and is unbiased, but also the researcher doesn't quite know for sure that the data is 100% true, as even if it is from the mutual response and the mutual viewpoint of the argument which you are looking into for the research, we don't know if the 100% reliability is there for the data. This could affect the research negatively, as it could make the whole test and whole investigation go completely wrong, as one small mistake in data can completely affect the rate of how reliable and how true your results are. Moving on to the types of data now, which is very situational for what you are looking for. Discrete data is a type of data used in surveys, tests, and other qualitative measures. As you can see, it's when we are looking at the days of the week or how many even numbers there are in a certain test result, or looking at the amount of students who are male or amount of students who are female in a class. This data is countable, as you can only take distinct values. So it would be, you can't have a half a student, so you can have to round it up or round down to the certain amount of students there are, because you obviously can't have half a person. And this is countable, whereas continuous data is measurable. Discrete data is mainly presented in a bar graph and mainly comes through easier tests and easier research methods, as the results are less in depth and are more focused on what the test is wanting to find out. Continuous data is a type of data used in measurements, timing, and weight. For example, when looking at the sprint speed of a competitor, that would be continuous, as the times can vary. Also, looking at the height of a human, this could change, so that means that this is continuous data. Also, you can have decimals within your continuous data, which would mean the data would be continuous due to it not being a strict value like discrete data. And also, this continuous data can be represented in histograms and charts such as that. Another type of data is ordinal data. Ordinal data is the type of data which is collected from surveys, which answers being such as happy, ranging all the way to very unhappy. This data is where the answers of the survey are unclear between what ranges between them, so we don't know what ranges between unhappy and very unhappy in this instance. And the data is laid out in specific order, from happy being at the top, from very happy being on the bottom, with results and choices being in between, where the participant wouldn't quite understand the differences between the sets, so they lay it out in an order clearly understandable from this instance, the happiest to the very unhappiest. In contrast, interval data is a type of data which is also collected from surveys, but with more numerical answers such as 50, 60 and 70. This could be when looking at temperature, 50 degrees, 60 degrees, 70 degrees. It is when the participant knows the difference between the answers, and it is still laid out in numerical order, but there is a clear understanding between what follows between each of the numbers, whereas towards unhappy and very unhappy it's not as close, as you can't really, if it was just happy and very unhappy, you wouldn't quite understand what would go in between. But with 50, 60, you would know, obviously 55 goes in between, and it says clear difference between the numbers. Ratio data is almost exactly the same as interval data, apart from the fact that the ratio has a true zero figure, allowing for a larger range of results. Examples of a ratio data include weight, height, and duration. The zero figure allows a larger range of results, as it the results can be more varied from all the way from 0 to whatever the highest number is, and whereas interval data doesn't quite have a 0 result, as it would confuse the participant and it could impact the results of the research methods. Moving on to types of quantitative research now. The first type of quantitative research method I will be talking about is questionnaires. These are when the researcher sends out questions to a set of people they are researching in order to gain anonymous information in a clear, easy to read way. As I've mentioned anonymous, this can help the research and the whole investigation massively, as people during interviews like to keep information to themselves, as they maybe be too embarrassed to state some answers and think it may negatively affect them. However, if the answers are anonymous and the questionnaires are anonymous, then that means that the people should be more open and more truthful when stating their answers. Questionnaires involve open and closed questions. However, for quantitative research, you are more likely to see closed questions, which are questions such as how many times have you been for a run in the last week, whereas open questions are more like why have you been for a run in the last week, whereas open questions are more suitable for qualitative data as they involve opinionated answers, as however closed questions are just statistical and straight to the point answers, which quantitative research wants. Some pros of the questionnaires is that they are direct questions which are clearly linked to the topic, and you cannot go away from the subject during quantitative questionnaires, as the questions quite clearly state what you need to answer. However, the cons are that you don't 100% know if the information is true. As the results are anonymous, people may be lying about what it is just as a prank or anything like that. So we cannot predict that all the information is 100% true. However, we would like to think that most is true, as it is anonymous and people like to be more open during anonymous questioning. The next type of testing is lab-based testing. It is used to collect information which requires scientific knowledge and and equipment, and a more in-depth approach. An example of this would be to find the amount of red blood cells in a patient which is being tested. The lab-based testing is very in-depth and very reliable. As you can see with the pros, it's true results every time and unbiased, as you cannot argue with science and you cannot argue with scientific results. However, the cons of lab-based testing is that it's time-consuming, as the tests have to be repeated over and over again to make sure there are no anomalies in the results, and it requires scientific equipment such as treadmills and controlling the variables without getting any external factors from putting off the person being tested. The last type of quantitative research method is field-based data collection. These are the data collections that take place in real-life settings, such as a football pitch, a classroom, or an office. So, for example, how different teaching or coaching methods affect students' or players' behaviour. These are very easy to set up and easy to understand what is going on. This is also like observation, yet these are more about how the data is obtained through finding results from the methods and students' behaviour. However, this is less efficient than lab-based testing, even though it is easier to set up, as it is harder to control variables and external factors may play a big role in affecting results, such as impacting behaviour and other affecting results.